The second topic that we'll be learning is morphological and differentiating characteristics of microorganisms. In this chapter, we'll be learning about the structure of different types of cells like eukaryotic cell and prokaryotic cell. Then we'll be learning about the different types of microorganisms, their structural characteristics, and their functions. So, to begin with, let us try and understand what are living organisms. The characteristics of the living organisms, as we all know, are reproduction, uptake of food, excretion, response to environmental changes, and susceptibility to mutation. These are the basic characteristics which makes the particular thing a living thing. So the question arises when we discuss about viruses, whether viruses are living or non-living. So at the end of this chapter, we will learn whether the viruses can be classified as living or non-living, and whether viruses can be called as cells or not. There are millions or billions of microorganisms which are existing in nature in different habitats. Like they are existing almost everywhere. The common habitats are soil, water, animals, animal body, dust particles, plants. They are existing in different microorganisms also. Apart from that, microorganisms are also present in some extreme environments that we cannot think about the existence of life in that particular habitat. What are these extreme conditions? Extreme cold conditions like Antarctica, extreme conditions like mines, coal mines, metal mines, where the conditions are not normal, where the temperature is extremely high or extremely low, the pH is extremely high or extremely low. So, under such environmental conditions, also microorganisms are existing. First, we'll learn about the classification of all the living organisms, both microorganisms as well as macroorganisms. So the classification started in between 18th and 19th century, where all the organisms were placed into only two kingdoms or two classes. These classes were Plantae and Animalia. It was classified by Carolus Linnaeus. Linnaeus is the other early systems of classification were thought to be wrong because they were based on inaccurate information. So that time the science was not very much explored. So many organisms were about to be discovered. So due to this limitation, as the microorganisms were not discovered, wrong classification systems were placed, and thereon when the microorganisms or the other organisms kept on discovering the classification system, it kept on modifying. That will be learning in the next slide. So researchers discover more about the physical and chemical characteristics of the organisms, and therefore we have the latest accepted classification systems. Let us have a background of major classification systems, old as well as the new, which is currently in use. These classification systems involve kingdoms and different examples of organisms which are classified under these kingdoms. So it was first in 1733 Linnaeus who divided the organisms into Plantae and Animalia. The examples under Plantae described were bacteria, fungi, algae, and plants. Under Animalia, protozoa and higher animals. But nowadays, we know that this classification system is rejected. New classification system is accepted. I am telling you about the old classification systems. Another classification system that came after Linnaeus is Haeckel's classification system, who further made three kingdoms based on the discovery of organisms: Plantae, Animalia, and Protista. So he added one more kingdom to this list. Plantae included multicellular algae and plants. That means this was the category for photosynthetic organisms. Animalia, animals, and Protista, a newly introduced kingdom, was for the inclusion of microorganisms that included bacteria, protozoa, algae, and fungi. But later on, this system was also dissolved due to the discovery of more living forms. So, the most accepted classification system is Whitaker's classification system, who divided all the living organisms into five kingdoms. That is why it is also called as Whitaker's five kingdom classification system. It was discussed in 1969. It was described by Whitaker. These five kingdoms are Plantae, Animalia, Protista, Fungi, and Monera. These two more kingdoms were added due to the discovery of bacteria and photosynthetic bacteria. Now let us see what are these different five kingdoms. First one is Plantae. All the multicellular large size algae and plants are classified under Plantae. So this is the category for photosynthetic higher complexity having organisms, that is multicellular algae and plants. However, there is a difference between algae and plants, that plants have vascular system, whereas algae there is absence of vascular system. Second kingdom is Animalia, that is animals. All the animals are classified under Animalia. All the organisms, microorganisms like protozoa and single celled algae, they are classified under Protista. The another fourth kingdom is fungi, which describes molds. And the fifth one is Monera, that includes all the types of bacteria. So we'll be learning in detail what are the characteristics of these five kingdoms which were discussed under Whitaker's five kingdom classification system. Another classification system which came after Whitaker's is Wu's classification system. This system it divides all the living organisms into only three kingdoms, that is Archaebacteria, Eubacteria, and Eukaryotes. So here in this classification system we can see that all the eukaryotic organisms, including microorganisms and macroorganisms, are classified under eukaryotes. For example, protozoa, algae, plants, fungi, and animals. Whereas WHO's classified bacteria into two categories: Archaebacteria and eubacteria. Archaebacteria are those which are existing in extreme environments, and eubacteria are those which are existing in normal environmental conditions. So we'll be studying in detail about Archaebacteria and eubacteria. So currently we follow Whitaker's five kingdom classification system, as well as for bacterial classification we follow Wood's classification system. Characteristics of Whitaker's five kingdom classification system. So what are these five kingdoms classified on? They are classified on the basis of these characteristics: cell type, cellular organization, mode of nutrition, cell wall, reproduction, motility, differentiation of tissues, and the examples of microbial representatives. So these are the five kingdoms: Animalia, Plantae, Fungi, Protista, and Monera. So the first characteristic difference between these kingdoms is cell type. So Animalia, it is eukaryotic, Plantae eukaryotic, Fungi eukaryotic, Protista eukaryotic cellular structure, and Monera kingdom, it has prokaryotic cellular structure. We'll learn more about eukaryotic structure and prokaryotic structure in next few slides. Second characteristic difference between these kingdoms is cellular organization. Animalia, multicellular. Plantae, multicellular. Fungi, multicellular and unicellular. Protista, only unicellular. And Monera, only unicellular. So the characteristic of Monera becomes eukaryotic multicellular, Plantae eukaryotic multicellular, Fungi eukaryotic multicellular and or unicellular, Protista eukaryotic unicellular, Monera prokaryotic unicellular. Mode of nutrition in Animalia are ingestive. Some exceptions are there which follow absorptive type of nutrition. Plantae, photosynthetic. Fungi, absorption of nutrients. Protista, these organisms, they absorb the nutrients. Some ingest and some are photosynthetic. In Monera, the organisms follow or they utilize the nutrients by absorption, and few of the organisms from Monera are photosynthetic. Cell wall is absent in Animalia, it is present in Plantae. In Fungi, it is present. In Protista, it is present. In algae. In Monera, it is present. There are few exceptions in Monera as well. Reproduction. So in all the organisms, asexual and sexual reproduction exists. Motility, in general, Animalia non-motile, Animalia motile, Plantae non-motile, Fungi non-motile, few are motile, Protista motile and non-motile, and in Monera both the types, motile as well as non-motile. Motility means ability of an organism to move. Then differentiation of tissues, that is complexity of tissues. Of course, in Animalia it is present. Plantae differentiate, different types of tissues are present. Fungi, it is present. In Protista, differentiation of tissues or specialized type of cells are absent, and in Monera also differentiation of tissues are absent. Microbial representatives that are classified under Animalia are non plantae, non fungi. Microbial representatives are molds and yeast. Protista, microscopic algae and protozoa. Monera, bacteria that includes eubacteria and archaebacteria. This is the five kingdom classification system of Whitaker's, which is the basis for classification of all the types of living organisms. Now we'll see the structure of plant cell, animal cell, and we'll compare that structure with the structure of bacterial cell. Mind that plants and animals are eukaryotic organisms. So they are eukaryotes. Bacterial cells, bacteria are classified under prokaryotic organisms. So they are prokaryotes. So here we'll learn about the difference between structure of eukaryotic cell and structure of prokaryotic cell. A representative of eukaryotic cell are plant cell and animal cell. Representative of prokaryotic cell is bacterial cell. So let us see what are the characteristics of plant cell. So in plant cell we can see the outermost layer is cell wall. Then in the cytoplasm, the membrane that follows after cell wall is cell membrane. Then in cytoplasm there are several inclusions. The most important one is nucleus. Nucleus is a compartment in which genomic material is present, that is DNA is present, that is chromosome is present inside. So it is present in a sac. That is because of presence of a nuclear membrane. Therefore there is a compartment called as nucleus. Followed by nuclear membrane, there are presence of Golgi bodies. Followed by Golgi bodies. Before Golgi bodies on nuclear membrane, there is presence of endoplasmic reticulum. Endoplasmic reticulum, Golgi bodies are involved in protein targeting. Then other organs are mitochondria, presence of chloroplast, starch granules, and the most important, ribosomes. Ribosomes are present at two locations, one in the cytoplasm and another in mitochondria. Ribosomes which are present in cytoplasm are of ATS type. Ribosomes present in mitochondria are of 70S type. You need to remember this. Second one is the structure of animal cell. So animal cell, you can see that the cellular shape is not fixed. That is due to absence of cell wall. That is the characteristic of animal cell. Rest of the organs, like it contains nucleus, nuclear membrane, endoplasmic reticulum, Golgi bodies, mitochondria, and few vacuoles. These are commonly present in animal cell. So these are the few characteristics which are shared by animal cell and plant cell. What are these? Presence of nucleus, presence of nuclear membrane, presence of endoplasmic reticulum, presence of Golgi bodies, presence of mitochondria, and presence of ribosomes, that is 80S type of ribosomes in cytoplasm and 70S type of ribosome in mitochondria. All these are the characteristics of a eukaryotic cell. This is the structure of a prokaryotic cell, that is bacterial cell. This structure shows the presence of cell wall, then cell membrane, then in the cytoplasm you can see in the cytoplasm presence of this nuclear material. That nuclear material is covalently closed double stranded DNA. You can see here the differences between eukaryotic cell and prokaryotic cell. In comparison, prokaryotic cell is very simple. That means the cytoplasmic inclusions are very less. There is no nucleus, no nuclear membrane, no endoplasmic reticulum, no Golgi bodies, no mitochondria, no chloroplast. So these are the characteristics of a bacterial cell. These are the characteristics of a prokaryotic cell. In addition, bacteria may have flagella. If you are talking about the ribosomes, the type of ribosomes present in eukaryotic cell are 70S in mitochondria and 80S in cytoplasm. In case of bacterial cell, mitochondria is absent, and in cytoplasm only 70S type of ribosomes are present in bacterial cell. So that is the major difference between prokaryotic cell and eukaryotic cell. Prokaryotic cell contains only 70S type of ribosomes. Eukaryotic cell will contain both 70S as well as 80S type of ribosomes.